Actually, first let me thank you for that kind invitation and several lovely days here in Paris. There's a great experiment going on around the world. It's an experiment that is centered at places like the Broad Institute in Cambridge in Boston. It's centered at the Beijing Genome Center. It's centered at a large number of sequencing facilities. And that great experiment is to look at all of the tumors that the people get, all of the kinds of cancers they get, different types of cancers, and to sequence the genomes, the complete DNA sequence of the genomes from the normal tissue and from the cancer tissue, and then to line up those differences and ask the question, which we have an inherent faith in the hypothesis, ask the question, do any of these mutations in the cancer contribute as driving mutations to drive the cancer, and can we learn from those mutations what drugs to use? That's the first premise, but also drugs to use that would be really targeting that particular cancer. But also, can we learn the prognostic outcomes of these cancers? What are the dangerous cancers? What are the less dangerous cancers? What are the ones that will metastasize? What are the ones that will kill patients? And what are the ones that patients survive with? There are many questions that the hope is that the sequences of the DNA will be helpful in teaching us those lessons. And this comes, of course, you could see from the morning session, comes, of course, from the fact that there are genes like the P53 gene, which play a central role in human cancers. And it's the sequencing experiment which shows it in a wonderful way. I have here, this is Magali's data, she's right here. And I have here a compilation of those sequencing data, right? So this is it's called the skyscraper plot for obvious reasons. And it represents three axes. The first is, let me say, you're not supposed to read any of the axes. And it's an overall picture of the skyscrapers. It's like coming to New York City. You enjoy the scene. We have 36 different tumors on this axis. We have 125 of the most frequently mutated cancer genes. We have abiding faith that these are genes that can cause cancer. I think the best evidence for that is that if you make mutations in these genes, or you eliminate these genes from animals like mice, that the mice will develop cancer over their lifetimes. So this is a direct inherited predisposition in this case, and in this case, most of these mutations are not inherited, but somatic. They rise over a lifetime of the patient and give rise to the tumors. So here's 125 of the most frequently mutated genes. And then this is the somatic frequency, the frequency of mutations you see in the tumor, but you don't see in a germline. We don't see in the genetic material we inherited from our parents. So these are mutations that arise over a lifetime, presumably because of our exposure to carcinogens and mutagens and bad luck in this particular case. Now, let me just say very, very rapidly, that rather than look at the details, overall, there's a sort of general picture that emerges. There are a group of genes which I'll call 3 to 5%ers. The genes in this area represent the 3 to 5%. They're not present in all the tumors. They're present in some tumors, but not all the tumors. And they represent a series of genes which are rarely found in cancer, but when found, they're important drivers of cancer. Oddly enough, these are the genes that the pharmaceutical industry has targeted so far for drug therapy, and so they respond very dramatically in drug therapy, and so again, giving evidence that they're important driver genes, they drive the cancer, but they don't make a big impact on overall cancer frequency and response because they've been targeted. They're targeted because they're easy targets. We know how to make drugs against them, essentially. That's essentially the difference, I guess, between the choice of these targets. Occasionally, some of these hit. That is to say, there's one called BRAF. You can see it something like here, right, where it's at low frequency, but there's an occasional tumor which has a very high frequency, so BRAF is in 50% of melanomas, but only 7% of colon cancers, right? So there's that. The first message here is tissue specificity, and there's some places where these drugs are very useful. Then there's a second group, which I'll call the 30%ers. They're represented by oncogenes like RAS, by tumor suppressor genes like BCL2, and MYC we heard this morning about MYC, and MYC is one of those genes. And these 30%ers are really important. They're at 30% in a large number. They're the higher guys in this group all the way across, and sometimes they're found in, clustered in many kinds of tumors. But there are no drugs that have been made against these yet. The PI3 kinase is one of these, and there's the first drug that's just been approved for leukemias and lymphomas, the gamma delta PI3 kinase inhibitors. And then there's a third group, which Magali has very generously pointed out, which here sits here down at the bottom under the letter P for P53, which is very common in many, many cancers. It's on average 50% of all cancers, but it has a very wide ratio of expressions in different tissues. For example, 100% of ovarian cancers have P53 mutations. About 80% of lung cancers, about 75% of colon cancers have P53 mutations. So the very big cancers, or many of the very big cancers, about a third of breast tumors have P53 mutations. They all fall in a class called triple-negative breast tumors. So there are very strong type and tissue-specific effects here. There's a tumor called the testicular teratocarcinoma, which virtually never has a P53 mutation. And fortunately, because of that, never having it, they can treat that tumor with cisplatin, a DNA-damaging agent. And you heard this morning that DNA-damaging agents are a stress, and that P53 levels rise and it kills the cell. And so here's an example of P53 in therapy, a good thing, actually curing it. And about 98% of testicular teratocarcinomas that have wild-type P53 could be treated and cured with the drug cisplatin. So this is a good example of understanding from the genes to the drug to the kinds of tumors that we have. But this is the ultimate, this slide is really the ultimate justification of why we would have a full-day symposium on this gene. It's a major, major player in cancer, and the question that everyone should be asking themselves is how does this gene function? What does it do? As you heard again this morning, it prevents cancer. That when it's lost in both copies of our genome, that's when it's most dangerous and gives rise to cancers. People who inherit a single copy mutation in a wild-type copy have a 93% chance of developing tumors over a lifetime. And so this is a major player in human and mouse tumorigenesis, and the question is, how does it function? What does it do? And you've heard us all morning, and we'll continue all afternoon struggling to answer the question, what does it do? It turns out to do almost too much, which is sometimes a diversion for cancer and sometimes a really exciting intellectual exercise to move in different directions. Okay, so this is, you see many versions of this as an introductory slide, what we think P53 does, but it's not enough to explain all of prevention of cancer. It's responding to a wide variety of stresses, and we heard about telomere stress, we heard about metabolic stress, we heard about a number of stresses, including even oncogene activation. A RAS mutation we heard this morning will activate P53 and prevent tumorigenesis or modulate down tumorigenesis. That's P53 sensing the RAS mutation, the oncogene mutation. This works, almost all of the stresses work through the ubiquitin ligase, MDM2, which negatively regulates P53, so that turns the ubiquitin ligase off. And this is a post-translational modification, and the half-life of the protein goes from, in the extreme, lymphocytes 6 minutes, up to as long as it keeps accumulating. So that means by 12 minutes it's doubled, by 24 minutes it's quadrupled. It's a very fast way to respond to stress. Post-translational regulatory way. And then it has a variety of transcriptional outputs which can lead to things like apoptosis, senescence, cell cycle arrest, all of which, when they were found, were reasonable explanations for the tumor suppressor activity, and none of which fully explains the tumor suppressor activity, so that we still struggle to ask the question, what are the ultimate functions of this? Because that's an interesting question, we decided to attack it in a slightly different way. It's possible to breed mice that have no P53, and they will develop tumors. They characteristically develop thymic lymphomas, so in the thymus, where the T cells normally develop, their lymphomas occur with 100% frequency. Right? And the thymus and the T cell, you'll see in a minute, represents a real opportunity to ask some questions at the cellular and molecular level that no other tissue gives us. So let me just show you what happens in these mice. This is the age of a mouse. This is the survival of a mouse. And long-lived mice here are wild-type P53. And so that's the lifespan of this strain of mice, which happens to be C57 black 6. A single mutation in one of the two alleles shortens the lifespan, and that's characteristic of a tumor suppressor. And these mice die at an earlier age. Mutations in both alleles shorten the lifespan further and move it to a very young age, and that's characteristic, again, of a tumor suppressor. So this shows exactly what inherited predispositions to cancer in humans will show, that the frequency with which you develop a tumor and the age at which you develop it are affected. The younger ages, you move to the left in this curve of age, and the frequency to higher and higher frequencies. Right? Now, the type of tumor that develops here is about 65% lymphomas, and because they occur in the thymus, they're T cell lymphomas. So let me tell you a little bit about the development of T cells in the thymus. T cells, for those of you who may not know, are part of the white blood cells that we have. They develop, start in the bone marrow, they move to the thymus where they further develop and become T cells. And one class of T cells, they're called CD8 T cells, one class of T cells are killer cells. And actually in cancer, they recognize cancers as foreign, and they often kill cancers. Right? And there's a whole new immunotherapy approach to activate those CD8 T cells to kill cancers. Obviously, cancers that are successful have paralyzed the immune system in this case. The other kind of T cell are called CD4 T cells. I'm making that distinction because you'll see in a minute these kinds of names. And they are helper cells. They really help the whole immune system make antibodies and make T cells, and they communicate. They send out signals to communicate between cells. So this is the history of a T cell. It comes out of the bone marrow, which is here off the slide to the left, and it moves into the thymus, where it has a particular set of markers so that we could recognize it as just the very first T cell to move into the thymus. It happens to be called double negative 1 or DN1 type cells. And at this point, a process begins that gives every new T cell that's born in the thymus a zip code. It gives it a marker, which is an individual marker. So every T cell that comes into the thymus gets a different marker on its surface. It's a receptor, and that receptor gives it its specificity for killing. So we have in our body almost 1 billion, actually about 450 million T cells, and those T cells have lots of different specificities, and it protects us against all of the things that will come at us over a lifetime, whether it's infectious diseases or cancer or otherwise. And the specificity of that protection is encoded in the information that makes the receptor. Now, you're not born with that. Well, you are born with it, but you're not conceived with that. We have no specificity at conception. So what happens is there are a series of genes, 3 genes in particular, called V, D, and J, which all move around, recombine, and make a different specificity in every cell. And there's a second component called terminal transferase, which adds nucleotides in, so that every receptor is a unique sequence, and that unique sequence makes a unique receptor. And that's why I call it a zip code. It's literally a place where that tumor could, that T cell could act. So we're watching here this recombination, which is called the T cell receptor combination, and we're just looking at one of the protein chains called the beta chains, and the first two genes to join up are DJ. That's diversity in joining genes, and that's DN2. And then VDJ joins up, so that's the three genes joining up to give you the receptor. That's a DN3 cell. And here's the beta receptor on the surface of the cell and a little bit of a fake alpha receptor. And then there's a P53 action that goes on. We actually saw, if you make too much P53, we saw this morning, the thymus will dissolve. It will, the apoptotic function would dissolve it, but sometimes P53 works normal and physiologically here at this checkpoint, but you finish the beta selection and get alpha and beta chains on the surface. By this time, this cell has receptor specificity, complete receptor specificity. Now, the cell, because it does this blindly, this recombination, actually has a very interesting problem. It has to be able to tell self from what's going to be foreign. And it does that by mating with self in the thymus. So there's a lot of epithelial cells in the thymus. It's watching for what you are, and those clones are eliminated. They're killed off and eliminated, and you don't have autoimmune diseases, and all that remains are the things that are possibly foreign to you, right? And that all happens within this DN4 phase. And then something happens which puts these magic proteins CD4 and CD8 on. That's called a double positive cell. And that double positive cell differentiates finally into a CD8 killer and a CD4 helper cell. And these leave the thymus, they go into your spleen, they go into your lymph nodes, and they reside there for life. So these T cells are born as you're young, they're made into all these opportunities for the future, and they reside almost for life over your whole life, waiting to be called forth. Now, they're called forth by a foreign antigen. It could be influenza infection in your lungs. And when they see an antigen on the surface of a cell and a receptor, they are triggered to divide. And a single cell will divide clonally many, many times. So we know that you've had an infection by just counting the number of T cells you have, right? However, I could, for example, determine how many new cells are born in your thymus at any time, right? Just by taking the DNA from the thymocytes, from the cells in the thymus, and sequencing across the VDJ region. It's only 200 nucleotides long. So what I do normally is put little primers on either side. I PCR up those primers. I PCR up between the primers, and I sequence all of the different sequences of T cells, right? And when I do that, the number of DNA sequences I get is proportional to the number of T cells you have in your thymus. So a T cell that's never replicated will give me one sequence. I'll call that a unique copy. A T cell that's replicated could give me 1% or 0.5% of the sequences that I get, and I know that it's part of a clone that's replicated up. What is a lymphoma? Well, it's one that goes to 2% or 3% or 5%. It keeps replicating, it replicates at a proportion to reality, and it takes over all the T cells. So I have a way, just by counting the number of reads of DNA in the thymus, to tell cells that never replicated, cells that replicated just right enough, and cells that are replicating too much, the tumor, right? So this is an example of such an experiment on a normal mouse, with a normal mouse, right? So these are numbers from normal mice. This is your thymus, or this is the mouse thymus, I should say. This is the mouse thymus, and here there are 138,528 unique clones. That means there's lots of DNA clones, there's lots of cells that have not ever seen an antigen in the thymus, that have not replicated up. They're getting ready to leave the thymus and go into your lymph nodes. And here are examples of the percentages of other clones. The largest clone is 0.03%. There's another one, 0.03, 0.02, and it goes all the way around, right? And this gives me, since there's no lymphoma in this normal mouse, this gives me the background for the normal mouse. The spleen is where these cells will go, And they go into the spleen and notice that there's about a 10-fold increase in numbers in the spleen. The largest clone now is 0.5% of the total. That means that, for sure, has seen an antigen. This is an animal that's about 3 weeks old, right? So the mouse has seen antigens. There have been pathogens in the mouse colony. It's made some T cells in response, and they reside in the spleen. So it goes up to 0.5, and there's 0.5 and 4 and so forth around. But that's the background in the spleen. And then if you really have an infection going, they'll leave the spleen and go into the bloodstream, where it goes around your body and it fights the infection. And notice that in the blood, there's one clone that's 3.5%. Right? So that means that our mouse colony had some organism that we've never been able to recognize, but the immune system has recognized it, and it's a way to tell us that there's been something in the mouse colony. Probably someone walked in the mouse colony with a human pathogen or something. And the mouse recognizes it, then it goes to 0.9, 0.6, 0.3, and so forth. So that gives us the background level. These are the normal levels, as it turns out, in a mouse. That gives us the background level that we would look at, right? So I'm going to show you two tables now of what happens in a P53 minus mouse to ask the question, do we ever get above these numbers, and what kinds of tumors do we have, right? And these are just looking at total unique clones. Now, in this case, I'm just going to give you the number of reads that we had, just to give you a flavor for what we do. We busily count the number of DNA sequences we find. And since age matters, right, here, we look at an embryonic, this is embryonic 17 days. Remember, Laura told you that the gestation period of a mouse was 20 to 22 days. So this is an embryonic mouse. It has not been outside of the uterus of the mother. So there are 2 males and 2 females. Because the immune system is sexually dimorphic, we wanted to see if there are any differences. There aren't, as you'll see. But here, somewhere between 31,000 and 43,000, 44,000 reads is in the wild-type mouse, and about the same is in the knockout mouse. So that would say there's no evidence for a lymphoma, because these are the unique clones. There's no evidence for a lymphoma in this mouse. At 3 weeks, about the same. This is called deep sequencing. That just means we've tripled, doubled, or tripled the number of sequence reads we take. The reason you stop is there's statistically no reason to go on past a certain number. That the statistics is good enough at this point. And secondly, it's expensive to go on for a number, so higher number. But we just do it to see. But here's the variation. It's about the same. 6 weeks, it's about the same. There's no difference. And here, for the first time at 9 weeks, this number is holding between 33 and 55, but this number's dropped to half. That's a tip-off. Something has happened in the knockout mouse. There are less unique clones, and that's because the number of clones in a thymus stays constant. And if you have less unique clones, we're going to be looking for clones that have started to replicate and crowd them out. That's what happens in the cancers. And by 20 weeks, for example, we have 36 to 52 in the normal mouse, and we're down to 800, as low as 800 to 5,000 in this mouse. And so, for sure, the normal clones are being crowded out, and the immune system's being compromised by a tumor. And you can see that by, instead of looking at unique clones, we're now going to look at the top 2 clones in terms of numbers. Right? So again, at E17, the percentages vary between 0.16 and 1.7. Right? They're about the same for the knockout mouse. It drops a little bit at 3 weeks, but it's about the same for the knockout mouse. 6 weeks, they're about the same. By 9 weeks, remember we had our first indication of a drop. We see 2.9%, 1.2, 21%, 13%. This is statistically significant over the reads. That tells us the first time we see a lymphoma, cells that are replicating with the same sequence, right? Here's a 21% of all of the clones are one sequence. They're one zip code. They came from one individual T cell, right? That tells you that that's a lymphoma. And by 20 weeks, the numbers grow. That is to say, they go to 70% or 43% and so forth, while the control stays. So we now have a timeline of the development of this, and this is going to mean something very interesting. The lymphomas are not occurring as soon as T cells are made. They seem to take about 9 weeks to be able to see them, and that's because there are additional mutations that have to occur spontaneously on this background of P53 mutation. So that we can conclude right away, P53 is not sufficient to cause the lymphoma. The loss of P53 is not sufficient to cause the lymphoma, but it's necessary. And secondly, there are additional mutations that must occur. And I'll show you those mutations. Okay, so here's the 20-week old clonality. This is 2 males, 2 females, 70% and 28%. And after that, we're down to background effects in the thymus. So 2 clones are dominating these, but we can conclude for sure that these tumors are not monoclonal. That is to say, they did not arise from 1 cell. They arise from multiple cells, and then they compete with each other for who's the winner, and even at this late stage, there are 2 winners, but one's better than the other. And the same thing is true with 43, 19, 5,000, 14%, 8,000, 5%, 1%, 1,000, 1.77, 70%, 5%, 4%, 4%. So these are oligoclonal tumors. That's the reason 100% of the mice develop the tumors. It says that this tumor suppressor is really important. You're not only developing, this mouse is not only developing a tumor over its lifetime, it's developing multiple tumors in the thymus over its lifetime. And that means there are a lot of independent events leading to the tumors. That's already a hypothesis that suggests that there's something about the rate of mutation occurring here which is higher than normal by a long shot in the absence of P53. Okay, so let's look at, now I'm going to actually try to make a little distinction. Rate of mutation is per generation. We can't count generations. We can estimate generations, but can't count them. When we look at a tumor, all we do is we sequence the tumor and we see a frequency of mutation, right? We can count all the mutations, and we call that a frequency because we don't take into account the number of generations it went through, right? So I'm going to stay with the term frequency because it doesn't make any assumptions about the number of divisions that rate would. This is actually from the Broad Institute. It's a very nice compilation of the sequencing of human genomes. And what it's measuring is the different kinds of tumors. That's like we saw in the very first slide, the different kinds of tumors. And here it's measuring the somatic mutation frequency, just counting the number of mutations that occur. And all these mutations are just single nucleotides, single bases in the DNA that have changed. And here are the changes. You can see C to T, C to A, C to G, T to C, T to C, T to G. You can see the kind of mutations that are changing, right? And over here on the right is melanoma. And we'll hear actually in the last talk about melanoma. Melanoma is the tumor with the highest mutation rate. And if you just take the average of the, you can notice that, by the way, tumors don't come in one frequency. They come in a distribution of frequencies. And so each point is a tumor, and it shows the distribution. But if you take the 50% point, you can see that there's about 10 somatic mutation frequencies per megabase of DNA. Megabase is 1 million bases of DNA. There's about 10 mutations per million bases. And that goes all the way back down here to rhabdomyosarcomas, which are childhood tumors, which are 0.1, right? So there's a 1,000-fold difference, or 10 or 100-fold difference between a rhabdomyosarcoma and a melanoma. And this is common. Some tumors have very high mutation frequencies, some tumors have low, right? And frequency, which we could get right away from the sequencing of the DNA of the tumors in a mouse, right? You'll find sits right in here in the middle, where breast cancer and pancreatic is. It's about 1 somatic mutation per megabase. And so P53, the loss of P53, is not raising the mutation frequency so high of point mutations, of these kinds of mutations, right? Single base changes. It's not raising it so high that it's very abnormal, right? So that isn't the mechanism by which P53 surveils. It doesn't really surveil point mutations very, very well. On the other hand, you could look at a different kind of mutation. These are called CNVs, or copy number mutations, right? Sometimes in the DNA, there's a loss of information, a deletion, right? And that loss of information I'll represent by red, right? And sometimes there's a gain of information, that's a duplication of information. Sometimes it's 10 copies even of the same information, and that's a duplication, right? And I'll represent those in green. And here are the 20 chromosomes of mice, and what you can see is there's some deletions, some amplifications in chromosome 1 where there's lots of green. There's a deletion of a whole chromosome 2. There's a deletion of chromosome 4 and 5. In fact, you could do this for 10 or 15 or 20 tumors. We've done it for about 15 or 20 tumors so far. Chromosome 5 is always deleted. It's commonly found in every tumor that one of the 2 copies of chromosome 5 is deleted. That tells us there's some loss on chromosome 5 that's contributing, if it happens every single time, right? Not true for some other examples. And then what's really characteristic of loss of P53, whether it's a human tumor or a mouse tumor, is found in chromosome 3 here. Look at this. There's amplifications of various sizes. This is a high, low, high, and then a deletion, and then an amplification, and then a deletion, an amplification, then a deletion, and so forth. And this alternating pattern of high mutation rate of copy number variations across the chromosome has come to be called chromatripsis. And what it is 5 minutes, okay. What that chromatripsis is in fact the recombination of 2 chromosomes so that, in fact, mutations and deletions and additions are occurring all over the chromosome. That is characteristic of P53 in human and mice. Okay? So here's the number. So if we want to have a quantitative feature that comes, the frequency of point mutations is 1 per megabase. The copy numbers that you see are changes are 276 to 422 over 15 tumors or so. And when you add that up, it turns out that that's very high. That's not common. So P53 is surveilling under normal conditions these genomic rearrangements, and in the absence of P53, it tolerates genomic rearrangements. And then the number of transformed clones, that means the number of cancers that arise per day, is 0.13 to 0.8 per day. At the extreme, that's almost 1 per day between the 6 weeks to 20 weeks. There's 1 transformation event a day almost, and that tells you that once these tumors get started, they're very diverse, right? So let me finish up by pointing out some things that are really remarkable about this. These are very diverse tumors, and yet, if we look at another locus that we see has a copy number variation, it has a deletion, right? That's a loss. It's called P10. It's a tumor suppressor gene in humans and mice. 88% of the clones in this female have the same deletion. 97% in this male have the same deletion. 85% in this male. And yet, in these 2 males, for example, there are 4 or 5 different tumors. So it's not 97% giving rise to 1 tumor. It's 45% and 20% and 10%. So there are 3 tumors, and they all have the exact same deletion. That means the deletion occurred very early in development, right? Before the rearrangement of the VDJ region, the T cell receptor. So all possible tumors have the same deletion, even though they arose later on. And that tells you there's an order to these mutations. The order is not, the mutations occur randomly, the selection occurs in an order. And the order is as follows. It's always P53 first, it's always P10 second. Let me give you the clue that the function of P53 is genomic stability. The function of P10 is metabolic instability, the use of glucose in very large amounts. And then there's a cell cycle mutation called cyclin D, CDK4, and then there is a differentiation mutation in the Notch pathway, which is a pathway that T cells need to give rise to the tumors and be T cells, right? So these mutations occur in an order. They're selected in an order. This isn't the first study that found an order. Vogelstein years ago found an order to the mutations that occur in colon tumors, right? They were APC, they were RAS, they were P53 at the end. There was also a BCL, I think it's BCL2 in the middle. So in that regard, there are now a growing number of examples that if you inherited a predisposition to a cancer, that the order in which these mutations are selected for may be preferred or fixed in this fashion. I'm going to now, I got 5 minutes, 2 minutes, 1 minute, 3 minutes, okay, good, okay. So I'm going to introduce you to Li-Fraumeni syndrome and just say something about humans. Rare autosomal, and this is the work we've done with Pierre Hanot, who's here in France. A rare autosomal dominant mutation. You inherit one mutation, you have wild type, but it acts in a dominant fashion. 93% of people, or 90% of people develop cancer over their lifetime. Young age of onset, that's just like the mouse. Lifetime risk, 85 to 90%. And in the data set that we've used, which comes from Iraq here in Lyon, there are 634 germline mutations. Families without mutations of P53 are not included. There are 2,483 cancer cases, over 30,000 person years and follow-up. So this is statistically very robust. And we come to a number of conclusions. I'm going to take you to one of the most interesting conclusions. This is estimated excess risk of cancer in the germline TP53 mutation carriers. So this is the age of a carrier, right? And this is the estimated excess risk. Excess is over the normal population, right? So you can see when you're young, you have tremendous excess, 100 to 1,000-fold excess in the young years, and it falls with time. And really quite remarkable, by the age of 60 or so, it actually falls below 1%. Right? In other words, the risk, if you manage to be in the lucky 7% of people carrying a P53 mutation over a lifetime and have never developed a cancer, right, you actually have a lower risk than the general population. The general population, by the way, is going up with age dramatically, exponentially. And the final thing I'd just show you about human cancers that are really quite remarkable is that the tissue specificity in Li-Fraumeni patients is completely determined by whether the tissue is replicating or not. So these are the tumors that occur in childhood, right? And this is choroid plexus papillomas, or tumors. Choroid plexus is a replicating tissue in the brain. Medullary blastomas, replicating tissue. Rhabdomyosarcomas, adrenal corticocarcinomas. These are the replicating tissues during childhood, right? This is breast cancer, the replicating tissues during the adult years, right? And in the elderly here. So this is the frequency at which you see those tumors. This hump is all due to breast cancer in females, right? And so the second thing to say is that there's a pattern to the tissue specificity in these patients, just as there was in mice. The first tumor is a lymphoma, but this isn't the lymphoma. This is always tracking the tissue which is replicating, which implicates strongly surveillance over stem cells, right? And then the final thing to say, and now that will be the end, and I'll be happy to take some questions, is this curve, which says that the kinds of tumors that we see somatic mutations in, remember I said ovarian, 100%, doesn't occur in Li-Fraumeni patients at all, right? I've said lung tumors, 80%, lung tumors are rare in Li-Fraumeni patients, right? Colon, 50, 70%, they're rare, right? Mesenchymal tumors, common in Li-Fraumeni patients, rarer in human populations that don't have the mutation, right? So there's tissue specificity impacting this pattern that we see in a very strong way. So let me stop here, and I'd be more than happy to take questions. Thank you.